started scuba diving about 40 years ago. The main reason being I actually had a fear of water where I couldn't see the bottom. In order to overcome that fear, I took up scuba diving and found it was absolutely amazing. The combination of being weightless and able to move in 3 directions provided an entirely new feeling that I was never used to. And in those moments of weightlessness and being able to move in 3 dimensions, my brain completely relaxes. Yeah, I'm Terrance Snutch. I'm a professor in the Michael Smith Labs at UBC, and I'm also the Director of Translational Neuroscience in the Javad Malfaghanian Center for Brain Health. A major issue with understanding and treating brain disorders relates to the fact that there are so many checks and balances built into the brain. There are both negative and positive feedback mechanisms. There are both inhibitory and excitatory circuits. As a result, combined, there are very few single-point failures in the brain. We focus on a family of proteins in neurons that let calcium into nerve cells. Calcium is a very, very important second messenger and controls everything from neurotransmitter release to excitability to gene transcription. And we wanted to understand exactly how these calcium channels perform all these different chores. What motivates me right now is the fact that these calcium channels that we've discovered underlie a variety of neurological disorders, underlie congenital migraine, cerebellar ataxias, essential tremor, various epilepsies, congenital deafness, congenital blindness, autism. Mutations in the calcium channels that we cloned, they provided new targets for both academics, biotech, and pharma companies worldwide to both study the underlying basis of many, many brain disorders and to develop new treatments to target these. 2 of the calcium channels that we cloned and characterized turned out to underlie schizophrenia and bipolar, 2 very serious human brain disorders that require new treatments, safe treatments, and effective treatments. Well, my first mentor was David Bailey, professor at Simon Fraser University, and David really gave me the opportunity to get into science. And up to that point, as an undergraduate at SFU, I had no real idea what research science was all about. He provided ideas, he provided resources, and then he let them take those ideas and figure out on their own which would be the best ones for their own career and their own interests. I took the same approach with my own lab and have been very blessed with excellent trainees over the many years who've gone on to develop wonderful careers in science and industry and in medical fields. The Margolese Prize is a wonderful and amazing recognition of my lab's work from the past 30 years, especially since it's adjudicated by an international committee, and it's always wonderful to be recognized by your peers worldwide. What excites me the most is the opportunity to translate our work from cloning calcium channels to understanding what they do in the brain to designing and developing new treatments for very serious brain disorders, and to get new drugs into human patients and hopefully to help them with their lives, their quality of life. It's the most impactful path that we've been able to go down.